talk about part of the documentary in this lab. So Norasquinoids and grapes. How do grapes develop their aroma? So if you guys still remember, last week there was this awesome seminar about wine labeling and wine quality. But before I start my whole talk about my research work, I just want to make sure that we're all in the same mindset. Is that for all of us here, wine quality does matter. And so for wine quality, if we want a good quality wine, actually everything, it starts from a good quality grapes, right? Okay, and for good quality grapes, what do we mean by that for a good quality wine? It could be like anthocyanins, the color of the wine, and definitely tannins, the phenolics. But there's also a very important part of it is the aroma. Right? And for aroma, it's actually, okay, so for aroma, we all have compounds that can give us a perception of the aroma. They actually come from a very wide range, a broad range of compounds. And they can be subdivided into various different groups. I'm just here to show you a few samples. So the first here on the left-hand side, I'm showing is ethiopia, which is a volatile ester. It gives us the aroma of strawberries. And on the right-hand side, dimethyl disulfone. It's the famous molecules at high concentrations give us an aroma of garlic, onions, but at a very low concentration, it actually gives us a perception of passion fruits. In the middle here, I'm showing the structure of beta-inone, which is in the Norasquinoids groups. It gives us the aroma perception of floral or fruitiness. And the main part of this whole thing is that all of these different chemicals are actually presented in grapes. So that's why we get all these different aroma notes from the wines. And in the wine world, basically there's intensive studies on the sensory of these aromas. But what I'm trying to point out here is, let's step back and look at this whole thing at a different one perspective. So first of all, the grapes need to produce these molecules. Then we can proceed. But how do grapes produce these molecules? Right, and I know me, like everybody else, who want to know everything about like all these different subclasses of these aroma molecules. For my talk, basically we'll be focusing only on the Norasquinoids. Okay, so how do grapes produce Norasquinoids? To make it in a nutshell, basically Norasquinoids, they come from carotenoids, which is also a group of compounds. They are presented in all different kinds of plants because they play a role in the photosynthesis and some anti-oxidation activities in the plant. Okay. And for the past decade, plant scientists have been working on how Norasquinoids are produced in plants, and they found out that it's actually the enzymatic reaction is actually done by a group of enzymes called carotenoid cleavage dioxygenases, and they use an enzymatic step of cleaving carotenoids into Norasquinoids. Here, giving an example, so beta carotene, which we will give the color of carrots, the orange color. So it was found in maize and many different other flowers that when there is CCP1, what it actually can do is that it does a cleaving step at the carbon I, carbon 10 valve. And after cleaving it, it releases the size of it, which are beta ions. And as I mentioned, carotenoids are a group of compounds, which it was pretty amazing that it was found that CCD1 actually can use all these as their substrates and producing the corresponding Norasquinoids, which most of us give us different aroma notes. And to make it a little bit more complex, CCDs are a family of enzymes, which in grapes, besides CCD1, we also have CCD4A and CCD4B. And still, to make it even more complex, it was found in other plant species that the CCD CCDs, they express differently throughout the developmental stage of the plant. So it's a very unique and complex system, and we want to try to figure out what's going on within the grapes. So as I mentioned, the genes, they express differently throughout the developmental stage. But let's step back for a moment and think about it. So for developmental stage, what do we mean by developmental stage in plants or in grapes? Or if we state it another way, how do we measure the maturity of the grapes? As we all know, grapes, they don't mature at the same time point. Basically, different vines, different clusters, a different facing side of the vine, and even down to like every individual berries, they don't develop in the same time point. They don't all reach maturity at the same time. So that's a pretty. So if you take a bunch of samples together and measure, you're actually measuring the mean of the measurements. But when we're taking like a bunch of samples, pulling them together, what we're actually doing is that we're actually masking out a lot of delicate information from each individual berries. And for my part of my work, basically we're dealing with volatile compounds. We're dealing with the genes that's related to these volatile compounds. Volatile compounds are not the major part of the berries. Volatile compounds are produced in a very low amount, and so are the genes that are related to them. So when we're pulling samples together, there's a tendency that we might be masking out a lot of information that we could get out of it. Which pulling samples is definitely a pretty typical or general method that we use for many of the biological experiments, because now we get a mean value. But at the same time, we might be losing out a lot of other information. So, is there a more precise way of making these measurements? Okay, so with this in mind, basically my research, what I'm trying to deal with is first, so how does the expression of the CCD correlate with spread development? And the second portion of it will be what are the substrates for the CCDs and grapes, substrates and products for the CCDs. Alright, so first, let's look at the first part. So how do we measure CCD, how do we correlate CCD with the grape development? And so basically, the variety that we chose was Thompson seedless. We all know Thompson seedless is not a typical wine grape variety, but the reason why we chose Thompson seedless is that grapes are unique, and actually Thompson seedless is the only one so far that are more easily genetically able for manipulation, meaning that we can do more molecular biology techniques to work on this, like we do gene knockout and gene overexpression, which so far was not able to be done on other varieties like Shiraz, Noir, or Cabernet. So this is the reason why we chose Thompson seedless, in the sense that there is a border range of further research. And what we did for the sampling was that we did a random sampling with single berries. So we're actually taking every individual berry as one biological sample that we take. What we did was that, so this is a part diagram of an instrument invented by Dr. Shackles. You might hear more about it when Aaron's in his presentation. So what we did was that, basically this machine has a probe and it measures the diameter and the firmness of the berries. And what we're using is actually the firmness. And afterwards, we cut the berry in half. For one half of the berry, we do the RNA extractions, and then we try to measure the gene expression of CCD1, 4 and 4B by serine analysis. And for the other half, we want to do is do volatile extractions pretty unique system because we can directly get to the gene, genetic information, and the volatile profile from one individual variable. And I know Wimberry you can't use Wimberry to evaluate the whole vineyard or evaluate the whole population of Thompson or the whole population of grapes. So we did a massive amount of sample on this part. And for the volatile profile, the instrument that we use is a GC-MSMS. The GC is gas chromatography. It's a very sensitive tool, and the detection system we use is the mass spectrum. Alright, so first I'm showing is that how the three genes they express throughout the time period. So I want to note is that for the gene expression data, all of the y-axis, they're all in log scale. So first we see that throughout the time period, CCD1 and CCD4B actually goes up. And because it's in log scale, you start from log 3 to log 6, it actually increases like 1,000 times. And I also did a Pearson-R correlation between CCD1, 4A, and 4B paired together. And we found that CCD1 and CCD4B actually have a pretty good correlation. But for CCD4A, it does not have a correlation with either CCD1 or CCD4B. And here I'm marking out this is the time period that could be Viridian for Thompson-Seeless. Thompson-Seeless is quite a variety. It's hard to pinpoint what Viridian is. And the reason why we don't have any data points before this was that before this point, basically each berry was like pea size. It's super small, and you just can't do RNA extraction from it. You won't get quantity or quality RNAs from that, and it would also be hard to do the volatile profile on this, that part of it. So we have samples over there, but we haven't done analysis on it so far. All right, so the second part is I try to correlate whether firmness and the gene expression can correlate with each other. Berries as they ripe, they start from very firm to less firm. Okay. And so this is the whole picture of each individual berries. It looks kind of messy, but if we only look at the part that is the firmness is below 10 megapascal, we kind of find a correlation, a negative correlation between the firmness and the gene expression. So this is actually data from whole samples throughout our sampling time period and from different vines. The R values don't look super convincing, but when we look down into one individual vine at one time point and from one individual cluster, for CCD1 and CCD4B, we actually can get a pretty good correlation between the firmness and the gene expression. But from this data that I'm showing, I have to also tell you is that we don't get this correlations from every individual vine. It's only some of the vines that we sampled from, we get this correlation, but some of them, the genes don't correlate well with the firmness. So a short conclusion or summary for this part. So first we observed the expression pattern for CCD1, CCD4A, and 4B grapes, and CCD1 and CCD4B. The expression patterns correlate with each other. And under certain conditions, the firmness can be related to the gene expression. And so for the other part of the berry, besides that, the half that we use for gene expression, the other part that we're planning to do the volatile compound is still in progress right now. So hopefully we'll hear it soon. All right, so right now I'll be switching gears to the second part of my talk, which basically we'll be looking at the substrate utilization of the different CCDs. Here I'm showing how the carotenoids are correlated with each other. Most of them are just one enzymatic steps from each other, and these will be the ones that we're looking at. so for this part, basically my experimental design, and we actually did with that. We are not looking directly in the grapes because basically grapes, they produce volatiles, and there's also all kinds of different molecules in the grapes. That could be a pretty high background noise compared to what we're looking at. So what we chose was we chose a biological system that actually does not have carotenoids. And we chose an E. coli system, which is a pretty general molecular biology tool that we use. It's been used for I don't know how long. What we did was that we introduced a plasmid that allows this E. coli to produce carotenoids to the different carotenoids that we just stated. And then afterwards, we introduce the CCD gene into the same E. coli. So right here I'm showing is an E. coli that can actually produce beta-carotene, which then the E. coli cells turn orange. And then when we introduce the CCD1 gene into it, CCD1 cleaves the beta-carotene. So the cell pellets color turn back to the original color of the E. coli. So that's why the whole essay is called the color complementation assay. So first, we can actually directly visualize if there's a difference, whether or not the CCD gene can work on the individual carotenoids. But we want to go further. We want to measure how the different volatile compounds are produced, and we also use the GC-MSMS to do the. So if there's volatiles in there. All right. So basically, what we did was we used 7 different E. coli and transgenic E. coli cell lines that we produced 2 different carotenoids. And for individual one of them, we transfer either CCD1, CCD4, and 4B, and we try to look at for our results. So here's a brief summary of what I got. So first, for the substrate utilization for CCD1, from the E. coli cell line that can produce lycopene, beta-carotene, and delta-carotene. What we actually found was that CCD1 can actually utilize these different carotenoids. And so for lycopene, for example, the complete steps over here, and it produced the corresponding arsenic 6-methylbihexene. For beta-carotene, it produced beta-inon. For delta-carotene, it produced alpha-inon on this side, and produces 6-methylbihexene on this side. What we also found was that for these cell lines, for CCD4A and 4B, they actually don't work that well on these substrates. They basically get a detection level that's close to control, which is the E. coli cell lines without any CCD introduced to them. So before I go further on, let's look at this structure. The interesting part of that, so CCD4A and 4B, they can't use delta-carotene, which the structure is with the alpha, with the epsilon ring here, produce the alpha ion, and over here the structure is there's a double bound over here. But there's also another carotenoid, which is relatively similar in structure, without this double bound here. It's called alpha zeta carotene. But with this simple change in structure, CCD4B actually gained its substrate utilization ability, which get a detection level that's higher than the background control, which it produces alpha ion and trans-strand nucleus. So a conclusion on this part. So of the carotenoids that we tested, CCD1 in general has a high utilization of the substrates. And for CCD1 and CCD4B, they actually have a difference in their substrate specificities. And from the substrates that we tested so far, none of them seems to be like a very promising substrate for CCD4A. And so what are the substrates? As for the first part of my talk, 4A is presented in the grades. If it's presented in there, there should be like some function of it. But at this point so far, we still don't. So there's this Chinese saying, there's so much that people want to thank, so we just thank God. But we're scientists, we have to be so specific. So first, I need to thank all my financial supports, the different fellowships and scholarships I got here. They make my life here in America much more easier than it would be. I also want to thank my advisors, Sue and Florence, which basically this project is a collaboration between Sue and Florence. And I also want to thank all the lab members within the two groups, especially so Anna and Ariel, regarding Sue's lab. They gave me very much amount of help on all the instruments that we're using, as you see, Speedy, the SMS. Without them, I don't know how long it would take to get my second part of my work that are given today done. And I also want to thank Mimic, who is in Florence lab. There. Basically, without her, I won't be able to find any chemical and anything in the lab. Like, oh, so where's this? And she's going to tell me. You should know better. And I want to thank Danella for. So this project was originally started by Danella, who's a postdoc I was working on this project. She basically built up all the bases. All the foundations of whatever that I'm able to present today, and I feel very grateful for her passing me a project that actually works. That's a good thing. And then I also want to thank everyone here that is listening. Thank you for your attention, and any questions? Don't know how much, but definitely there might be, that might be one of the issues. The decay of volatiles, basically in every additional processing step that we use, there would be a tendency of decaying on the volatile compound. So what we actually, the only way that we can try to tackle this is that we use a control at the same time. So for all the volatile compounds that we do analysis on, basically we always have a control compared to the ones that we use. So they're all processed in the same way, and in the end we make it Yes.
When you got the barriers in half, did you know kind of the orientation? Because even within the barrier, there might be variations in the rightness, in the anterior posterior limits facing the sun inside of their cluster.
Yes, that's definitely one of the issues. So what we took to various,
Were we able to detect anything? Did you notice anything?
Okay. What I found was that. So basically, on the same line, the north-facing side of the vine and the south-facing side of the vine, they get different sunlight exposure. And from that, what we actually found was that for the south-facing side of the vine, the berries that's took from the south-facing side of the vine, in general, their firmness is lower than the north-facing. But we didn't get down to detail like for individual clusters.
Sure.
Yeah. But my guess would be like the berries that's buried inside, it might be a little bit different from.
So the south ones were less firm, were softer.
But it could, besides the developmental and the stage of the berry, it could be just like hydration.
Yeah.
That could also be.
When you have the E. coli that's expressing or that's making carotenoids, then you have like, let's say CCD1 in there. Is the CCD1 on all the time, or do you induce that?
Yes. So when they made the vector, yes, the vector was designed so that the vector that contained the CCD gene is supposed to be inducible. But what we found from the vector was that there was a leak to it, which if you don't induce it over a period of time, the CCD1 still expresses. So for that part of the experiment, basically the way that we try to get around it is that we try to make everything for individual cell lines. We try to process everything exactly the same way. So that's the time point compared to OD to make sure that everything is sort of in the same